Okay, so let's talk citric acid cycle. First of all, cellular respiration will occur in 3 steps, and it is basically the pathways that will give us energy, and we'll use carbon dioxide we will use oxygen and produce carbon dioxide. So it occurs in 3 steps. The first step is so after glycolysis, we will produce pyruvate. We will produce pyruvate, and this pyruvate can then, as I said in the glycolysis video, it can either go into become lactate, or it can undergo oxidative decarboxylation and become acetyl coenzyme A, which is this one. So, and I'll talk about oxidative decarboxylation really soon. But this is the first step. Of cellular respiration, and it's really important to note that it's an irreversible step. Once we have acetyl coenzyme A, we cannot go back to pyruvate. That doesn't happen at all. So the first step is oxidative decarboxylation of pyruvate, and we will get acetyl coenzyme A. The second step of cellular respiration is the citric acid cycle. That will be the second step. And citric acid cycle, as it was written in our professor's slides, it was like acetyl coenzyme oxidation means citric acid cycle. Okay. And then the third step of citric acid of cellular respiration is the electron transport chain. And we're going to talk about this really soon, but I just want you to note that in this step, this one right here, we will have an enzyme, which will be the exact enzyme that acts in electron transport chain in a complex. So the electron transport chain has 4 complexes, and the second complex will have an enzyme that is exactly that is the enzyme in citric acid cycle. So that's important to know. Okay. Oxidative decarboxylation, which was the first step of cellular respiration. So don't forget, this step is an irreversible step. Also, there are some ATP calculations in this step. So why do we have oxidative decarboxylation? Because in order to enter citric acid cycle, we have to have acetyl coenzyme A. We have to have it. And acetyl coenzyme A gets produced by this process by the help of pyruvate dehydrogenase complex, which is this enzyme. And it's also this enzyme, like the yellow, green, and red is the enzyme. Okay, so where does this step occur? Oxidative decarboxylation. Okay, so pyruvate is produced in the cytosol. Then pyruvate will, so if we have the cell right here, and then we have the mitochondria right here. So pyruvate, and then it will travel inside, because citric acid cycle takes place in the matrix of the mitochondria. So pyruvate will travel to the matrix of the mitochondria, and here in the matrix of the mitochondria, we will have oxidative decarboxylation. So, what else did I want to mention? And in this step, pyruvate, right here, it has 1, 2, 3 carbons, will lose 1 carbon in form of carbon dioxide, and we will form a thioester bond, I think, or basically this one. Now, what's important to know about pyruvate dehydrogenase complex? It has 5, I don't know what to call it. It needs 5 cofactors, okay? And these 5 cofactors will be B1, which is TPP, and it will also need B2, which is the FAD. It will need B3, which is NAD. It will need B5, which is coenzyme A, which you can see here that it's been added to the molecule, like right here, that's B5. We can also see it right here. So where is it? Here. Yes. And also we will need a lipoate thiol group. And if any of these cofactors is missing, the enzyme will not work. And the enzyme has 3 enzymes, if that makes any sense. Okay. But that's basically what you have to know. And also, this enzyme is regulated. So it is inhibited by high levels of ATP, NADH, and acetyl coenzyme A, but it will be activated by high levels of ADP, NADP+, and pyruvate. Remember for calculations that we receive 1 NADH for this reaction, so we will get to 2.5 ATP from this reaction. So if we get a question in the colloquium where we have to calculate all of the energy from glycolysis until electron transport chain, then do not forget about oxidative decarboxylation, because here we will receive 2.5 ATP. Okay, good. Now let's talk about citric acid cycle. So it will have 8 steps, and it's a cycle because oxaloacetate, which is the product, can react with acetyl coenzyme A and form citrate again. So it's a cycle. So you can just read this. I just copy pasted from the lecturer's slides, but you can. I would just like to highlight a few things. So step number 1 is the rate limiting step. We will use citrate synthase, which is the enzyme that will attach acetyl coenzyme, it will aid in the reaction between acetyl coenzyme A and oxaloacetate and form citrate, which is the first, which will be the first, it will form citrate. And then there will be an isomerization of this citrate, and it will become isocitrate. This right here is an intermediate, but it will go from this one to this one. And this is together with the help of aconitase, which is a lyase. Also, I think you have to know this because they said this in the lecture, that we have to know if this enzyme is, aconitase, what kind of enzyme is it? And you have to say that it's a lyase. So that you have to know. Then we get to steps number 3 and 4, which is oxidative decarboxylation, and it will give us 2 NADH plus H plus. So this step and this step are important because these are the steps that will give us NADH. And also this step, malate and oxaloacetate, will also give us NADH. So the enzymes here that are working is isocitrate and alpha-ketoglutarate dehydrogenase, which is an oxidoreductase. Then in step number 5, we will get our substrate level phosphorylation, so we will get a GTP. This is important to know, substrate level. Then step number 6, which I talked about in the beginning. I mentioned here that this step right here would be connected to ETC and to a complex. That's because here in step number 6, we will have succinate dehydrogenase, which will oxidize succinate to fumarate, and in that process it will release FADH2, and that will be used in the electron transport chain. So that's important to remember. Then we will have step number 7, blah, blah, blah. And then we'll get to step number 8 and we will get NADH plus H plus. Now let's do some ATP calculations. When we do ATP calculations, we have to see the cycle or the pathway or whatever. And then we just have to remember one thing, that 1 NADH will give us 2.5 1 NADH will give us 2.5 ATP. 1 FADH2 will give us 1.5 ATP. 1 GTP will give us 1 ATP. So that means, just ignore this for now, that means we have to look at the cycle and we have to look how many NADHs can we get from citric acid cycle. And we look here, we see that there will be 1 NADH produced here because of the arrow that's pointing there. And there will be 1 NADH from this reaction. Also, there will be 1 NADH from this reaction. So basically, NADH will be 3. Which is what I've written here. So we will receive 3 NADH from citric acid cycle. And if 1 NADH is 2.5, that means we will get 7.5 ATP from only the NADHs. Then we go to the next thing. We will get 1 GTP right here. So that means 1 GTP, 1 ATP. Then we will receive 1 FADH2, which equals 1.5 ATP. So that means all of these together, if we add them, 7.5 plus 1 plus 1.5 equals 10 ATP. But that is from 1, so we will get 10 ATP from 1 acetyl coenzyme A, from only 1. But that means from 1 glucose, you will get 2 acetyl coenzyme A. So you will get 10 plus 10 ATP. So you will get 20 ATP from 1 glucose molecule in citric acid cycle. And yeah. Okay, so honorable mentions. Okay, so just like important steps, enzymes that you have to remember because they will be important for other pathways. So in step number 5, we will have substrate level phosphorylation and we will get 1 GTP. And also you have to know this in case, I don't know, but in case they don't give us the pathway, which they probably won't on the colloquium, if they don't give us the pathway, then we have to remember how many GTPs, how many ATPs we have received from the cycle. So therefore you have to remember these steps because these are the steps that will give you the energy. Then we have oxidative carboxylation in step 3 and 4, we get NADH and H. Dehydrogenation in step 6, we get FADH2 and in step 8 where we will get NADH. Also, in step number 6, we have succinate dehydrogenase, which is directly connected to ETC to the electron transport chain in complex 2. Then another thing that they mentioned in the lecture was succinyl coenzyme A, which is, where is it? This one right here. So you see here, succinyl coenzyme A goes through this reaction and it will form succinate. And as it does that, it will release energy. So the breakage of, breaking this bond will create so much energy that there will be a GDP just right here. And when this one breaks, when succinyl coenzyme A breaks its bond, it will release energy. And this energy is used to incorporate inorganic phosphate to a GDP and form GTP. So succinyl coenzyme A is important for that reason. And that's what I've written here. Now to regulation of citric acid cycle. Really, you can just look at the picture here and you can know, but activated by substrate availability, and I will just read this out now. Activated by substrate availability of pyruvate will activate the citric acid cycle, and it will be inhibited by product accumulation. Because if we get the products, why would we even need the citric acid cycle? So if we get a lot of ATP and ADH, that will inhibit the citric acid cycle. Also, this will not inhibit, but breakdown of amino acids can also activate the citric acid cycle. So we have amino acids, glutamate, glutamine, blah, blah, blah. If they get broken down into alpha ketoglutarate, well then alpha ketoglutarate can take part in the citric acid cycle, and that way we will activate the cycle. Then there's something here about isocitrate. So this step right here, when citrate goes to isocitrate, it's a reversible step, and isocitrate can be reversed to citrate. Citrate can diffuse out of the cell or out of the mitochondria and then enter the cytosol. And when it enters the cytosol, it will go to phosphofructokinase, which is step 3 of glycolysis. And it will inhibit glycolysis, because if phosphofructokinase is inhibited, then glycolysis will not happen. And so that can be an inhibitor. Another thing we have is reverse phosphorylation of the enzyme pyruvate dehydrogenase. Oh my god. Dehydrogenase. So reverse phosphorylation of pyruvate dehydrogenase. And maybe you remember that pyruvate dehydrogenase is the enzyme that is this one. Actually, I don't think I mentioned what the name was, but it's called pyruvate dehydrogenase. This enzyme can be regulated by reverse phosphorylation. So here we have the enzyme, pyruvate dehydrogenase, and it has 3 phosphate groups on it. If it loses one phosphate group, it will be activated. And if it's activated, then pyruvate dehydrogenase can convert pyruvate to acetyl coenzyme A, and acetyl coenzyme A can enter the citric acid cycle, and we can, So that is kind of a regulation of citric acid cycle. For the last thing, citric acid cycle and cancer. So if we get we have a lot of enzymes in the citric acid cycle. And one enzyme, for example, is the alpha ketoglutarate dehydrogenase enzyme. If there's a mutation in this enzyme, if this one gets mutated, it will lead to the accumulation of 2-hydroxyglutarate. And 2-hydroxyglutarate can go, and it is a competitive inhibitor for TET2. And TET2 is involved in histone demethylation. But if 2-HG through competitive inhibition binds or inhibits histone demethylation, this could lead to cancer, could lead to altered differentiation and tumorogenesis. Okay, so that was it for the citric acid cycle. Bye.